Yes. Oh. Hello, everybody. I would like to welcome you in the third lecture in MGBS, and today we have a very outstanding speaker, Professor Jutta Papenbrock, and I would like to introduce her in a few words. Prof. Papenbrock is a professor at the Institute of Botany in the Faculty of Natural Sciences, Leibniz University, Hanover, in Germany. Her research is focused on sulfur metabolism, especially at the molecular level and in abiotic responses. Her group was also interested in the investigation of stress-tolerant plant species for different purposes, utilizing the natural diversity of plants. Among her field of interest are the investigation of plants as biofilter for the biodegradation of xenobiotics, but also as sources for valuable plant-derived components and as novel food sources. Thank you, Dr. Jutta, for joining us and showing up on time today. And please go ahead for your presentation.
Yes, thank you very much for this kind introduction. Actually, I cannot see my presentation anymore. Can you see it?
Yes, we see it.
Okay.
It's very clear to us.
It's very clear, but just let me see what the problem is.
You can please start sharing the now again, because now we can't see your presentation.
You can't see it. Okay.
Yes.
So I see.
You can start sharing again your presentation.
I will do so. Okay.
Yeah, it's clear now.
Okay, but now you should be able to see it in presentation mode. So thank you very much again for this invitation. So today I would like to talk about new salt-tolerant crop plants or new salt-tolerant plants. And just as a short introduction, actually in the past we worked a lot also with Balanitis aegyptiaca. I think many of you from Egypt are very familiar with this plant species. It's also called desert date, and you can find it in many parts of Egypt in the desert. And I think that is also a candidate that can be used as already stress-tolerant plant, and the fruits can be used for many different purposes. So that was also a very, very interesting project with an Egyptian PhD student, and I really enjoyed this time with him very, very much. And currently, actually, a small plantation of Balanitis aegyptiaca is growing in the south of Egypt, and maybe we will have a kind of a forest of Balanitis. So again, that was a great project and is still going on. Yeah, so I work at the Leibniz University in Hanover at the Institute of Botany. And here on this picture, you can see the main building of Hanover University with this horse that is very typical for Lower Saxony, actually. So as you are all aware, there are a lot of agricultural challenges that we have, like rising sea levels. And due to these rising sea levels, we also have the loss of arable land, actually all over the world. Of course, maybe some coasts are damaged more or influenced, but almost all coasts of the world and therefore agricultural land are challenged. Also, we observe soil degradation. That's, I think, another major problem. Of course, there are also soils that are very, very much degraded, as you can see here on the slide, and others are less damaged, but actually can observe that soils become more and more degraded and therefore less fertile. And one reason is actually salinity. So an increasing salinity leads to soil degradation. To all these issues I just was mentioning, another contributes a lot. So we have the freshwater scarcity. And I was just discussing with Ahmed before the start of this presentation that he also thinks that even in Germany, that had a lot of rainwater in the past, more and more droughts are actually observed. That means climate conditions become more and more extreme. And therefore, less fresh water is actually available. And that, of course, also contributes to the fact that there's less crops can be irrigated, and that makes it more difficult to feed the growing population. And all these issues, we are faced with all these issues, and we have to find a solution for that. Here this example of soil salinization, where you can really see salt on the field directly. And it was actually observed also all over the world that the electrical conductivity increased over the time. You can see here on the x-axis the years, and you can see that in several countries like Australia, China, India, and so on, that there's an increase in electrical conductivity in many soil types, actually. So an increase in salinity. So there might be several strategies to tackle with these problems of salinity, or in general with the influence of extreme climate change, extreme weather conditions as well. And there might be two major ways. One is actually the domesticating of wild relatives of stress-tolerant plant that already possess all the necessary stress-tolerant genes. Or one could also try to incorporate genes from wild relatives into domesticated crop plants. I think both strategies are very valuable, and both are also very, very important. We focused more on those plants that are already stress tolerant and that might be domesticated over the time to act as a new crop plant and replace some of the crop plants that are already used. And of course, in parallel, one could also improve agronomical techniques and use, for example, biostimulants. I will come to this point only very shortly at the end of my talk, because I think also stress-tolerant growth-promoting microorganisms can also contribute a lot to plant health, to make them more stress-tolerant, and can support these kind of strategies that I was just mentioning. On this slide, you see some characteristics of glycophytes and halophytes. So most of the crop plants that you can see here on the left-hand side, that are the crop plants we usually grow on the fields. They cannot grow with high salinity. They only grow at relatively low concentration of sodium chloride, whereas the halophytes can actually grow also at higher salinities. So there's actually also kind of a salinity threshold, how you define glycophytes and halophytes. And you see already some halophytes, some naturally stress-tolerant plant. And I would like to talk or to focus on, in this talk, mainly on Salicornia, samphire also called, that you can find actually all over the world, at least in the temperate region. And in many parts of the world, people already try to grow Salicornia. But actually, the experience with Salicornia is still quite limited, and there are a number of open questions that need to be answered before actually Salicornia or other stress-tolerant plant can be established as new crop plant. So we have to think about cultivation systems, about agronomical techniques, fertilization, how to harvest these plants. Of course, we have to learn what is actually the yield and how the plants that we harvested are actually combined, what we can do with it. If you think in the bio-economy, it would be great if you can not only take some part of the plants to feed the people, but maybe other plant parts can also be used to feed animals as feedstock, or also for other aspects, for example, for the production of valuable chemicals. So I think that would be great if we establish new crop plants, that we could also think how these plants could be used in a complete way and not only focused as food. Of course, we also have to make sure that we really work with genetically verified and homogeneous species. For example, with homogeneous seeds, which is kind of difficult if crop plants are newly established. And we also need a better understanding of the molecular basis why actually these plants are stress tolerant. And for that, it's important to know more about gene expression and maybe also about genes that are of interest for breeding purposes. Finally, of course, you could also think about genetic engineering. Even stress-tolerant plants can be improved with respect to their stress tolerance, but of course also with respect to their tools to use them as crop plant. Yeah, as I mentioned already, I would like to focus in this particular talk on Salicornia. We also work with other stress-tolerant, salt-tolerant species like quinoa, for example. It's also a very important, promising plant species, but also with other plant species like Cryptoma maritimum, for example, and so on. But Salicornia, it's a species that is very well known for its high levels to exceed salt up to 1 molar sodium chloride. Just to remember, in the oceans, for example, the North Sea, you have 3.5% sodium chloride, 35 grams, and Salicornia can survive much higher concentration. It's a succulent plant, and that is actually the basis why it is so salt tolerant. It's an annual halophyte that you find, as I said, in temperate regions, in salt marshes, or in coastal areas in general. You can also find it in the inland on salty patches, so it can be also grown there. So I think it has high potential for applications in agriculture, can be used as a source for bioenergy and also for some pharmaceuticals. And actually, we were part of an EU Horizon project. This name is Aqua Combine, and there's also a nice homepage where you can find more information about Salicornia, but about this project, because here it was investigated how Salicornia could be used in a holistic way, in a bioeconomical way, so that all parts of the plant species have a value for the farmer. Okay, where did we get started? So we were first interested how Salicornia europaea actually respond to long-term cultivation under salt, both under salt deficiency and under salt excess. And for that, we try to identify genes that are involved in salt stress and that could be used to examine the stress reactions. So we usually we start our experiments growing the plants in a hydroponic cultivation to really grow them under controlled conditions. So we use different concentration of sodium chloride, as you can see here. And then we harvested the plants in 15-day intervals. Here you can see an example of the different salinities, and in the middle of the experiment, at the end of the experiment. And I think you can clearly see that here in this range between 7.5 or even 15 gram per liter sodium chloride, that they form the biggest biomass. And I think the biomass is actually the yield of Salicornia, because this is mainly used for all purposes. So this is summarized here in this graph, as I already mentioned. Maybe I should explain. Here on the x-axis, you see the days when we harvested the plants. Here you can see on the y-axis the fresh mass, and here's the legend to understand the bars. And from 0 gram per liter to up to 30 gram per liter sodium chloride. And as I already mentioned, that we have a maximum biomass production at about 15 gram per liter, as you can see here, after the different harvesting time points. So that seems to be quite an optimal growth for this halophyte, or growth conditions at the salinity. We also analyzed total phenolic compounds, because we were interested how the different salinities actually act on the stress level of the plant. And we usually investigated shoots and also the roots. And I don't explain it again, because I think the code is now already clear. And here you can see if the plant species were grown without any sodium chloride in the medium, they perform or produce less biomass, but they are also stressed. That means this species, Salicornia, is actually stressed when there's no salt. That means it's an obligate halophyte. It really needs salt for growth, for the production of biomass. Next we started to establish the gene expression analysis. There were already 1 or 2 studies published at that time, but it was not clear actually what kind of reference gene were used. In many publications, people use actually actine as a reference gene. So that is the gene all the expression of the genes of interest are referred to. And we were interested to establish a good system. Therefore, we analyzed a number of different reference genes and selected 3 of them because they resulted in the best values for our purposes. And then we analyzed the expression of some genes of interest, mainly involved in already known salt tolerance mechanism. As I was already mentioning, the Salicornia is actually a succulent plant species, and that means it accumulates in the vacuole high amounts of sodium, and this is an energy-dependent process. That means they are pumps in the tonoplast of the vacuoles, and so by this the sodium is actually pumped into the vacuole, into the plant, and then into the vacuole. And some other genes are actually necessary that are also all transporters localized in the tonoplast membrane. So this was already investigated previously, but not so much in detail of Salicornia. So here you see some results of the different transporter encoding genes. And here again, I don't want to go into very detail, but you see here the very obvious bars. The higher expression is actually found in those conditions where no sodium chloride was added. That means those genes that are actually involved in stress tolerance, they are also more expressed when there is no sodium chloride present. It was quite interesting, actually. So that is the shoot results. And here on the next slide, you can see the gene expression in the roots. Again, the same genes. And actually, there is not much activity visible. So no changes in gene expression over the long term of our experiment. So here's kind of a summary of these kind of results in a simple way. So we compared shoots and roots, and over the time, these lines are just drawn of the gene expression over time. And you can see if there's a lack of sodium chloride, there's actually higher expression of some genes involved actually in salt tolerance. In the optimum and also in the excess, there are not much changes in shoots and roots. If you check on phenolic compounds, it's similar. So when the plants are stressed, there's a higher formation of phenolic compounds, and the phenolic compounds increase with time, especially in roots. In shoot fresh mass, I already indicated that there's kind of a maximum at around 15 grams per liter or a little bit lower. So that was the first part of my presentation, and the take-home message is that the cultivation of obligate halophytes definitely needs, as the name says, a certain amount of salt. Otherwise, the plants are really stressed. And here we actually identified for Salicornia an optimum of about 15 grams per liter to produce fresh biomass. And this is actually the yield. It can be used as vegetable, as salad, as there are many recipes. And in some parts of the world, it's actually like gourmet food. For example, in France, people pay a lot of money for Salicornia. It contains a lot of vitamins, antioxidant compounds, and so on. So therefore, it's really a good source, a good vegetable source, and can be used as a everyday vegetable if there would be enough access to this vegetable. Actually, this remembers me. Here you see some hairy roots. Maybe you're familiar with hairy roots. So it's kind of a production system where you can use to produce a lot of secondary metabolites. And because Salicornia actually accumulates a lot of valuable secondary or specialized metabolites, we also try in parallel to establish some in vitro culture techniques. And we also try to establish a regeneration system of Salicornia so that we could also modify, do genome editing of Salicornia europea or other plant species. But for that, we first need to establish an in vitro culture regeneration system. So now I would like to extend this study a little bit, because actually there are several Salicornia species. In most of the publication, they actually deal with Salicornia europea. But actually there are many more, and actually their differentiation, their determination, it's very, very challenging to determine the correct species. And one of our, let's say, tasks that we are currently working on is to determine species by molecular markers, so to make sure that if we would like to use them for future breeding processes, that we actually start with a defined correct species. Because here in the next slides, you will see that actually there are big differences among the species with respect to their salt tolerance, but also with respect to their composition and also other characteristics. I think not all are really aware of these differences, and that is something what we are currently working on to show that it's really important that you know whether you work with species X or species Y, for example, with Saliconia europea or Saliconia ramosissima. So here, that is an experimental plot I would like to show some results from. So we have grown Saliconia europea and ramosissima at different salinities. So here we have chosen a similar hydroponic system, and we have grown them at 15 grams sodium chloride per liter and 30 grams sodium chloride per liter, and harvested them after several days. And here's just the fresh biomass shown. And you can see here, if you just compare two species at the same grown, cultivated at the same conditions, there are major differences with respect to the fresh mass. That means it definitely makes sense to check carefully what kind of Saliconia you would like to cultivate. Now you maybe could say, oh, it's just in hydroponics. No, actually, we have also made some experiments with cooperation partners together, and we also see major differences between the species also on the field. And what maybe is also an important good take-home message that it doesn't definitely make sense to grow that species that is naturally occurring at this particular region and in these conditions. Okay, so we have much higher biomass with ramosissima. We also analyzed the gene expression for the salinity-related genes. And there are also differences among these two species, which is quite interesting, because you could think of that maybe Saliconia, or within the genus Saliconia, there might be actually different strategies to deal with salt. That is something we learned from these experiments, but we didn't go deeper into this aspect. But I think it's also definitely makes sense to try to better understand how actually these different species deal with salinity in different ways. So the same is also true for other genes. I don't explain it again in detail because there might not have enough time for that. Here's kind of a summary, and that also shows differences in fresh weight and the species in both salinities. And also, just in summary, differences in gene expression in both species for the different genes related with salinity tolerance. So here you can also see the summary, and that means it is really important that if you're interested in the cultivation of Saliconia, you should maybe choose or select or try in pre-experiments which Saliconia species grow best, because they differ in their salinity tolerance and probably also in their physiological parameters in their adaptation to stress. Okay, so we extended this study. We actually investigated several Saliconia species, not only two, from all over Europe, or actually seeds were ordered from different sources all over Europe. We also included two other closely related species from this family. And we have also grown it in all these species were grown in our hydroponic system, as I described already. Here, Hoagland solution, 15 gram sodium chloride per liter. pH was set, so it was they were cultivated on substrate, and then the seedlings were transferred to the hydroponics. They were harvested in liquid nitrogen and so on, and they were then used for analysis by ICP-OES, which analyzes the elements, micro and macro elements. This we were interested, or we did these measurements because we were interested how actually the sodium chloride influences the uptake of other elements, because that is also an important aspect if you want to cultivate new crop plants. You have to know how to fertilize them, what is necessary to fertilize them on saline soils. And we also would like to analyze secondary compounds. This is actually still in progress. In addition, we also analyze the composition of the different Salicornia species, also with respect to better know whether they can be used as a food for animals, not only for humans, but also as a feedstock. So therefore, it's interesting how much protein, fat, fiber, ash, and so on they contain to control how and to control whether Salicornia can actually be used. Now here you see again the experiment of all the species, just early, mid, and late development of plant species. And here you see some results. Here are the different species are listed, and here you find on the right-hand side the abbreviations for these species, and you can see from these graphs. Again, I don't want to go into each detail, but here on the left-hand side, you can see that there is a big difference with respect to the biomass of the species under the same cultivation conditions in shoots at different time points. And you can also see here that the sodium concentration within the plants also slightly differ. So here's a difference in Sarcocornia fruticosa in comparison to the other plant species. And that actually changes also here over time. And here we analyze the water content, and you can see that the plants, of course, accumulate more minerals and especially produce more lignin, and therefore the water content actually becomes lower, but the water content is different in the species. That means there are differences among them. Here you see the total phenolic compounds content that is also different in the different Salicornia species and in the two additional species I was mentioning, Sarcocornia and Atrocnemum. And they actually also slightly differ in their biochemical composition. And without going into each detail, I just want to summarize that again, each species might differ in composition and secondary metabolites, and therefore it's important to choose the correct one that you are interested in. So here you see some pictures. So, and you can of the species we investigated, and here you can see also some morphological differences, but it's really hard to differentiate them just by morphology. And as I said, we established a system of molecular markers, also investigating ploidy level, and to really to be able to differentiate among the different species. Okay, I think, I summarized that already. And I think I have a few more minutes, because now I would like to switch to another species that is also highly salt tolerant. The interesting aspect of this species is that it shows a different tolerance to different sodium salts. So that is another important aspect. So you cannot find only sodium chloride in the soil, but in many parts of the world, you have actually different sodium salts. So here in this case, where this plant actually grows, you find both sodium chloride and sodium sulfate. And the effects of these two salts are different to the plant, and that is has an academic interest to better understand the influence of either sodium chloride or sodium sulfate, but also can be used if we would be able to identify the different strategies of this plant. It could be also maybe transferred, or some genes could be transferred to other plants, to other crop plants maybe, to make them not only tolerant to sodium chloride, but maybe also to sodium sulfate. So these pictures were actually taken in Argentina, in a salt desert in the middle of Argentina. And this is a cooperation project that is done with Professor Mariana Reginato from the university in Rio Cuarto, in the Cordoba province. And yeah, you can see here the plant on the natural localization. These are the fruits containing the seeds. And as I said, it grows quite well, or at least in comparison to other plants that contain high levels of sodium chloride and sodium sulfate. But if you grow them in the lab, they show different stress levels with respect to these two salts. So here you see again the experimental system with the seeds that are scarified, germinated, and then they are grown in Hawkland solution in a hydroponic system. And here you see some growth progress after 24 days and after 35 days. So we compared either the control with sodium and sodium sulfate, and a mixture of both salts, and harvested the plants after several hours after the last salt treatment was actually applied. Yeah, here you can see the procedure. It was very, very, very difficult to extract RNA from this plant, from Strombocarpus strombolifera, because it contains a very slimy substance, a mucilage, that makes it very difficult to analyze, to extract RNA, and also to analyze this plant species in more detail. But maybe it's also interesting to actually analyze this particular mucilage in more detail. Maybe it contributes to salinity tolerance. Okay, here you see the process of RNA extraction, and we also were finally able, after a long process, to analyze the genes that are expressed in the chloroplast in the plants that were cultivated in the different treatments. Here on the on the x-axis, you see the controls. Here you see the sodium sulfate treated plants. Here the sodium chloride treated plants, and the relative gene expression. And here you can already easily see what I mentioned before. And here are the different time points where we harvested. And here you can see that this transporter NHX actually was induced by sodium sulfate after about 24 hours, whereas sodium chloride did only very slightly induce the expression of this particular gene. That means with our molecular analysis, we may become to answer this question, what is the difference among these two effects of the two salts, maybe become closer because it was already investigated since many, many years, I would say even since decades, how these two different salts act on the plant. But so far, actually, only physiological data were obtained. So these are the first molecular data that will be extended maybe also to RNA-seq analysis to get a larger overview. But first, we wanted to make sure that we can really handle the RNA extraction, cDNA synthesis, and so on. So also some stress genes were investigated, also some transporter genes, and some transcription factors that are involved in salinity tolerance, and also some sulfate transporters, which are actually of high interest for me because I'm also very much interested in sulfur metabolism. And I think, as you can see from this small as part of the results, that we definitely have differences in the reaction to the different salts, and we will continue with that in more detail. Yeah, also a stress indicator I don't want to explain right now. So we could establish a qPCR analysis. We see differences among the genes of interest with respect to these two salts. And we would like to investigate this model plant in more detail, and maybe here we can use this strategy to transfer genes by genome editing, or to not transfer, to modify the genes of interest also in related plants of the Fabaceae family, and Strombocarpa strombolifera might be a good source for that. So I'm almost at the end of the presentation. I think I've maybe 2 or 3 more minutes, because we also are interested in plant growth promoting microorganisms, because our results, and of course also of many, many other people, I know that many people in the audience might have very good results. If they add plant growth promoting microorganisms to the crops, that they can increase the yield, because they increase plant health and reduce the stress. And but often it was observed that actually the microorganisms could not survive in the soil, and we try to use biochar produced by pyrolysis from different organic material and cover them with microorganisms. So the biochar has large pores, as you can also see here on the picture, and so we cover them with both fungi and bacteria, and we could see actually major effects on the field, but also in hydroponics. Here we don't understand the reason yet, but this is, I think, also a very promising aspect to increase stress, strategy to increase stress tolerance of plants. Here you can see where we treated quinoa under soil stress, under different salinities, which is given here, 0, 2, and 4 grams per liter. And we added biochar with microorganisms. And you can see that we, especially at the higher salinities, there's a big increase in fresh weight between biochar and microorganism treated plants in comparison to the control, and at the lower salinities the effect is lower or is not present. So obviously at higher salinities the plant growth promoting organisms can be covered on the biochar, covering the biochar, can actually mitigate the stress. In this experiment, I don't want to go into detail. We used, in this case, Tropeolomaius, which contains valuable glucosinolates. And here we could actually increase both the fresh weight in comparison to controls or in comparison to treatments with other, just with biochar or with plant growth promoting fungi. And also, and that was of high interest, we could also increase the yield of the glucosinolate we were interested in. So I think that is also quite promising that you can also influence plant metabolism by microorganisms and mitigate stress. So that is my summary. Yeah. There are still many open questions if you want to use stress-tolerant plants as new crop plants. Many steps to go, as you could see from my title. That means we have to better understand on the molecular level these kind of plants, but also define breeding goals and then step in this direction. Maybe highly stress-tolerant plants could be used to identify genes that could be also transferred into glycophytes, or maybe new strategy of stress pathways could be identified and then modified. Yeah, and in general also plant growth promoting organisms, microorganisms could be used also at least to mitigate stress to improve the soil and reduce the influence of salinity and other stresses on the plants. Yeah, here is the picture of our group in front of the institute and some funding agencies that support our work. Yeah, I don't know, maybe there are now, I'm open for questions, and if there are any, I'm happy to answer them.
Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow.